Hello, everybody. Welcome to our algae solutions for global challenges session. We intentionally made
it is a joke, huh?
It's not a joke. We believe in it. No,
it's not a joke?
That's why. No, it's not a joke.
It's a joke.
And we intentionally wanted to do it broad, okay? Because in 2 hours we will not be able to cover all solutions and all challenges, but we will try to present you some examples of some successful research and successful studies. And we are sharing it with Dina. We are working with Dina a lot in microalgae and fish. So in the first lecture, we'll be representing how a known study of algae can be incorporated with fish and how we can help by using these technologies, aquaculture technologies, to help in growing food demand. And I welcome Dr. Jose Pedro Canavate, whom I'm following really many years, but we never met. He's now working, he's a marine biologist, working a lot with microalgae and lipids. And now he's working in the Andalusian Institute for Research and Training in Fisheries and Agriculture in Spain. So, Jose Pedro, you're welcome, and please.
Okay, thank you very much. First of all, I want to thank the organization for the effort of bringing me here, especially for me because one flight was canceled due to technical problems, so I have to say thanks. And a special thanks to Ina, of course, for the invitation to the interesting conference. This, for me, I'm a marine ecologist. I'm working surrounded about still water, so for me, it's a really enriching experience coming to a dryland conference, discussing with you, showing you what can be done about the microalgae and global food supply. So first we have to ask what's the relationship between microalgae production, which is done in an aquatic media, can be performed in a wide range of places. So why? What relationship can be with microalgae, aquaculture, and drylands? The other important question for this talk is, can we really handle to use microalgae to increase global food production? So these two initial parts, I would like to price, first of all, current state of aquaculture microalgae and raise options for how to increase the global food production on the base of microalgae, with special focus on the integration microalgae production with use of dryland. That's it. Yeah, the initial answer I want to raise here. I will try to reason why we could use microalgae dryland. But there are many other drylands that are so. First of all, I also want to bring your attention to the huge genetic diversity of microalgae that you can see has been represented here, which is behind many questions we are going to see later on during the presentation, that all processing of microalgae, the use, and many aspects. As just to have an idea, the Holland plants came from this lineage, and all of the other microalgae in the globe were the result of secondary and tertiary endosymbiont processes, and they are behind different cell types and different biochemical composition, which explain or also adequate for different applications. That typical representation of microalgae application, the value and the volume of each application. But here I would like to focus only in this side, food source. I have also highlighted it as the functional, I would say the functional side. I will treat it tangentially because it's very difficult to differentiate sometimes the nutritional from the functional side. But anyway, I will do my best to focus only on the food source side of microalgae. Well, microalgae are supporting fisheries. This is not very well known for the science, but there's a lot of studies relating primary production in ocean with the fisheries resources in large ecosystems, in large marine ecosystems. But microalgae are better known because their use the aquaculture at the hatchery level. It maybe was the aspect that popularized microalgae use, but if we analyze it, we can see that this is only a qualitative side. So this is not a significant quantitative contribution to food supply because it's performed at very small level, but only contribute qualitatively. But we have what is called pond aquaculture, which was very smartly reviewed by Amit Neori 10 years ago, and he established microalgae as a very important support in pond aquaculture. This has not been acknowledged as should be, but I want you to pay attention to this because it's really important, this aspect. And we also have to consider that shellfish production relies completely on natural production of phytoplankton, which is not less. But totally, and according to FAO, we have microalgae supporting 70% of global animal production. Well, what we want is to increase this 70% with the use of different microalgae, using different systems, different applications, to support feeding various consumers. But first, we can think about human consumption, but we are not herbivores. So we are only at the level of a supplement. We are consumers as a supplement. That means that it's not a quantitative application in this instance. But if we consider trophic support for farm animals, even both fed species, livestock and aquaculture, and non-fed species, microalgae do play an important role, which can be partial or full covering. And depending on this tendon, we can see that the impact of microalgae, the quantitative impact, is increasing along this trend. Well, we all know that the global food demand is increasing. This is a statistic everywhere published, and that this demand has to be met in scenarios where we cannot fish anymore, we cannot use anymore fresh water, we should preserve forest, we must not deforest anything else, we have not to produce gas, greenhouse gases, and we have to avoid pollution. It's all fact constrained food production. So we are trying to produce microalgae, or we have to produce without increasing this 30% land use and of the 70% freshwater consumption. Anyway, so there are different activities that I will not consider here. Just improving the efficiency of current technology is a way, of course, being performed to increase food production. But here, I would like to find new net, what I call new net resources. I mean, not work on the already known resources, but new net food resources that can be suitable for feedstock. And in this case, key choice is what I call here high productive aquatic phototrophic systems with no freshwater footprint and no competition for land use. That means that we are going to work with marine species. We have not considered freshwater species at all. But we want to avoid any freshwater footprint. This is an option. Okay, phototrophic production is not new. It's not a new idea. It was raised in the 50s, late century, in the MIT, in the roof, the MIT, with freshwater species, but it was very difficult to upscale to the proper level. Okay? More recently, the production of macroalgae has been tried to associate with other activities which can interact synergistically with capture of carbon dioxide, with the use of wastewater, and also to recover some kind of system, in this case, systems. Have the opportunity to produce a highly nutritional feedstock to offset fish exploitation and forest deforestation. So we need a product based on macroalgae, which has nothing to do with increasing fisheries exploitation and forest deforestation. But the first question is how high nutritional value of microalgae. Sometimes we speak about the high value, but microalgae has their own position at the nutritional level. And just very quickly from the lipid side, we can see that microalgae varies a lot in their lipid profile, in the fatty acid profile. The essential fatty acids which are used for index, for nutritional indices, varies a lot between phylogenetic groups. And the level of most of the algae is far above that of plant oil level. But only dinoflagellates approach to a reference commodity as it is fish oil, okay, which is the target for producing acid. So we can see that no species obviously can be used for our purposes. On the contrary, the essential amino acid is constant throughout phylogenetic groups. And compared to land plants, we see that essential amino acids, both methionine and lysine, are well above the level land plants, but histidine is very similar. But once again, if we put livestock and fish, their essential amino acid profile is still higher. So we have a good product, but it's not a reference, it's not resembling the nutritional profile that we want for a high efficient feedstock. Microalgae have also, because that difference, a huge variability in the digestibility and viability. Their walls are very different, and some cells are very fragile, and others are very, very strong. And also, they have a strong intracellular protein carbohydrate interaction that make difficult the viability of proteins. And microalgae have also, although toxins and growth inhibitors are very little described in microalgae, at least in those that are used at culture level. But we have to, anyway, be aware and minimize possible side effects of toxins, growth inhibitors, and also contaminants, basically heavy metals. And what is more important is microalgae are cell single proteins, so they have a high free nitrogen competition, which is limiting. This limits their use in non-herbivorous species. So for higher trophic levels, it's the limit for using portion of macroalgae in feed. And finally, in this side positioning of the microalgae, we know that the functional side with some functional components, microalgae is increasing food conversion. That means that it may contribute as well to food supply by increasing the efficiency. But once again, this is expected to be a supplementary side because the inclusion levels are not very high in this system. We are dealing with the functional level. And so we ask which trends are being done. Well, aquaculture sector and livestock sector works basically with compound feed. And so the main option is to take the algae, put it in the feed, right, and see what happens. But I want you to bring this question. I draw this line, it was not easy. But this means that the way of processing microalgae cannot be the same that the way for using land soybean, for example. Microalgae are more sensitive, particularly more variable in their sensitivity to processing. And that means, for example, that during harvesting, we have to harvest a suspension of 0.1% volume. During harvesting and dewatering, we may have very different responses. Algae. We have some algae, this is Rhodomonas. Rhodomonas just look at this breakdown. And with other like Petacellus and Chloropsis and other cells can tolerate quite well centrifugation. But this is very important because if you centrifuge, you precipitate with this, maybe if you put this in the feed, then the feed does nothing. So the product is not adequately processed. This has to receive very little attention at the moment. Very much more attention in the future. Then we had the drying process. When we had the case, we had the drying process, which also affects losses nutrients. There are different ways of drying, drying, but this also affects the nutrients and, of course, as I said, translate the different properties in the compound feeds. And I insist this has been overlooked and must be considered in future research. Well, in spite of this, a lot of work has been done. Although the project concept was project in the early 90s by John Benneman. He visioned that the microalgae could be used in feed, but took 20 years to start taking off research in this field. And since 1910, a lot of studies has been done. This is a lot. There is a lot of redundancy in this kind of studies. I have selected these 3 reviews. It's a very recent review. If any of you are especially interested, you can see them and avoid to read the hundreds of reviews, similar reviews, which are the literature now. But the main conclusion is that between 5 and 15% is a suitable present inclusion level in aquaculture compound feeds. Because, well, as I said, the moment only whole cell without any specific processing is being used. And this relates as well with the trophic position of culture feeds. We low down with the system, reversal. We low down trophic position, we can increase. We review the paper, we can see that more algae can be put in the food, which is logical. Regarding livestock, some studies have also paid to the use of microalgae in livestock, but the problem here I see is that the aquaculture has replaced the consumption of, for example, fish meal in livestock. Now, at present, all fish meal is consumed by aquaculture, and maybe aquaculture will pay anyway more than livestock activity. And also, aquaculture species are more efficient at transforming proteins. So, I think microalgae at present for livestock is been a complicated issue. But anyway, the quality, the meat quality, has been also studied, could be a solution. But once again, this is a supplementary issue. So it won't represent significant quantitative contribution. Well, just to review this first part, we have to consider that we need to adequately process the biomass. We have to either protect or labelize, depend on the algae. The diversity is huge, as I said. Some species must be labelized to make nutrients available, but other must be preserved and protected, because otherwise they break down. In this way, we could increase some part, I think, of the proportion of algae in the food. And also, at the moment, cultured microalgae should be preferentially used in farms, animal diets in 10 different species, high value. Because there is still high cost, which do not allow for a competition with common feedstock. We have in many places that benefits of algae production, which are right, all of them are right. They have farm independence, more productivity, everything is right. But most reports considering this aspect do not consider that in the agriculture, there is other aspect important, like those one, which really outweigh the advantage of microalgae. So microalgae are like this, are more efficient, but if you compare with agriculture products, then there is some problem with the economy. There's a lot of system for aquaculture, for microalgae production, and depending on the system, we have different applications and also a strong impact on production costs. There's also a lot of studies considering production costs, and most of them consider this range between 12 and 69 euros per kilo dry weight. Those some are more optimistic, but the problem with most of these studies is that they are very, very theoretical, and they don't consider some very important issues, which are the upscaling problems, and also, well, the stochastic events that happen in outdoor conditions, especially the visitors, which are very hungry for algae. And, well, there's also a problem that it makes culture less stable. If you look for prices, you will see that most prices are within that range. Water. This is a search made on the web for bulk microalgae wholesalers. You see that most prices are the same. And even for new species, for new ring species, if the price may be even higher, around 100 euros kilo. For other applications, like nutraceuticals or cosmeceutical, price might be even higher because production is performed in more intensive systems. So, first, I want to just to check which are the best cost operation. I will go fast here. But anyway, we have nutrient represent around 0.45 euros per kilo. If we consider efficiency, well, approximately prices for carbon, nitrogen, and phosphorus. It has been tried to link cultures with flue gas, waste, water, but this, in this case, this direct use will restrict. So if we are going to use microalgae for food application, we cannot go to this system because food requires another quality of the food. Mixing is another issue. Mixing means that the water must move continuously. And well, if you look for published result, the minimum energy that must be put in the system is 5 watts per square meter, which is this. Now, we don't know the price is very valuable, but if we use a conservative value for kilowatt hour and normally accepted production, we see that we have about €0.50 cost for dry matter, which add to the previous. And in terms of energy, this means that the energy that we have to put is only
1.5. If we want
harvesting is also hard. But I will go fast because I want to go to the other side, which is if there are other options. Here is when we try to supply this use of nutrients by natural fluxes, continuous by ultra pumping, and machine by live organisms. Here we have to acknowledge that production will be lower, the amount of money to lower, but we can achieve system-based microalgae at large areas as required. Here is where I want to bring from the bench dry land and play the game. There are many types of dry land. We have the desert for cover, but we have a lot of dry land from human activities, which are huge. And what can be done, of course, filling and managing, of course, how and water sources we have to go anyway, coastline, sea, estuary, groundwater water, but we have to consider that cold water must contain inoculum for it. And a very rapid scheme will be this. We manage hydrology, nutrient to create a plankton community, which can be used to feed a low trophic level position in which we harvest continuously in a similar way that it is done in the ocean. You know, the ocean is being fished down for copepod, for palm oil. The only difference is that here we get an upgraded food resource without extra fishing, sorry. So we get new net production from marshland, for recovered marshland areas. And in the production, we have very little information, but from San Francisco Bay, we know that around 1.5 ton metric ton can be produced per hectare per day of forage fish, but we have to share it with wildlife because this is a system. Wildlife, and we can go to one ton. Just there in comparison, as I said here, we have this problem with fish meal is stabilized between 5 and 6. How much land we would need? We would need obviously about 5,000 square kilometers of dry land for production. This is a double inspired. We have to see that there are many areas and where. Well, there are recent inventories say that we have around this surface in the world, tidal flood, mangrove, and tidal marshes are this surface, and the original historical area is small, and that means that around this surface has been degraded, and well, what fraction would be sustainable? This is a big question now, and it's difficult to resolve in the future because a lot of factors has to be combined. Just as an example, in a big land salt marsh area in the South Spain, about 1,500 square kilometers, we can see that in the last century, only this 300 square kilometers has remained preserved. And we can estimate that only about 200 kilometers could be used for this reflooding and reuse. Similar has been driving other marshland areas in the States and China. Well, we have that about 30% of this total degraded marshland area could be rehabilitated for algae-based system integrated production, but question, would this be feasible? Well, we know from this period that 35,000 square kilometer mangrove were transformed in 2 decades. So, but maybe Tunisia when it turns to destroy and to recover. But anyway, we have to keep in mind this in the future and be ready. They have economic reasons, including value of the biomass and also the value provided by ecosystem services, especially that of nitrogen capture, which can be estimated between 4,000 and 5,000 euros per hectare, only that value, if we consider the value of water treatment plant, and also be adapted to probably sea rise level, which will let us to reconvert some low yield threatened areas, which are dedicated to agriculture, another water-based activity, which will improve anyway the resilience. Finally, just considering the desert, I will like to just consider that some initiatives have been proposed: seawater, evaporation of seawater, marine algae produced with seawater as well, and are used, but some limitations, of course, may exist, and that should be clearly sorted out. Permeability, orography, so on. In any case, a notion of opportunities is waiting for dryland research to increase and to have a new opportunities for microalgae based protection. So, thank you very much.
I know it was an exciting presentation and very interesting, and I'm sorry that we don't have time for questions. And so everyone welcome to contact Jose Pedro and ask him questions. And today is the last day of the conference, so we will a bit patient. And so next I would like to invite Professor Yoav Livney from Technion. He's from Biotechnology and Food Technology Department, and he will share about using macroalgae. We're moving to macroalgae in food technologies, the protein and the starch source. And
thank you very much, Dina. Thank you very much, Dina. Thank you for the organizers for inviting me, and happy to be here. So yes, the topic of my talk is macroalgae as alternative source for protein and starch for food. And this work has been done mainly by my former PhD student, Meital Katsir, and was in collaboration with the lab of Dr. Alvaro Israel from the Israeli Oceanographic and Limnological Institute, Haifa, and Alex Kozel from Tel Aviv University. So as you all know, the world is expected to have 9.7 billion people in 2050. And as you all know, there's growing shortage in land, especially arable land, dwindling freshwater supply, and climate change, and great loss in biodiversity. So there's a great burning need for new and renewable, sustainable sources for food. Again, loss of less and less water, fresh water, and the eating, the use of animals, particularly beef, for food is not sustainable, and we really must find ways to stop that and or at least minimize that, not only because of animal welfare, just because we cannot sustain that anymore, and also because of health reasons. There's importance to transfer to a healthier lifestyle and use more plant-based, vegetable-based food. Both sustainability and health reasons. And so the oceans and the seas are becoming an interesting direction for potential solution, although we have to do it in a sustainable and responsible way. One suggestion is to develop offshore seaweed farming as the way to grow food, new food sources on the sea. So eating macroalgae or seaweeds is not something new. That's been done for ages, and in some parts of the world still a significant part of the diet. However, the use or the development of components, ingredients from microalgae like proteins, starch, is relatively new. The main ingredients that have been extracted from algae are the indigestible polysaccharides like gums, alginate, stuff like that. But protein and starch from algae is relatively a new approach. Seaweeds they have a relatively short growth out cycle. They have very minimal requirements. And they don't need fertilizers. They could help address the land water scarcity crisis. Their utilization can also help counteract the climate change. Overall, there's mainly three groups of macroalgae: the brown, the green, and the red. And their nutritional composition on a dry matter basis have about 30 to 50% carbohydrates, about 7 to 50% proteins, about 7 to 25% minerals and vitamins, the group V vitamins, lipids 1 to 2%, but especially the omega-3 are interesting, and phytochemicals like phenols and carotenoids, about 1%. If we look at the various components, particularly protein, you see the red algae have the highest amount of protein, followed by the green. The green and the brown has the least. Also, other components of interest. Algal proteins are a good source for essential amino acids. There is variation between species, of course, and with seasons. Algal carbohydrates can be again split into indigestible like carrageenan and alginate ulban, and the digestible like starch. So we'll start with the algal proteins. As I mentioned, they're up to 50% of the dry matter, and they have a good amino acid profile, beneficial potential health effects. And so we would like to check whether macroalgae can serve as alternative sources for proteins in our diet. Algal glucans, first of all, the green algae have starch, which is very similar to terrestrial plant starch. Red algae have a slightly different starch type called Floridian starch. And brown algae have a glucan called laminarin, which is digestible. The first main species we worked with is the sea lettuce or ulva. It's an edible seaweed. It's a good source for protein, and it's got a high content of polysaccharides, up to 25% starch, very high growth rate. The other type we worked with is Gracilaria. It's a good source, even better source for protein, higher content of protein, and also a good high content of polysaccharides, but mostly agar. Ulva grows very fast, high starch content, but it grows during the colder season mainly, while the Gracilaria, which has a higher protein content, grows mainly during the warm season. So we can have like in a combination of both, we can have an optimal year-round production of protein and starch. So the goal of our research was to utilize macroalgae as a sustainable source for protein and starch, keeping in mind the zero waste vision, try to minimize byproducts or waste products, use only food applicable extraction processes, and to look at functionality after the extraction. So the specific aims were to develop a food applicable protocol for the extraction of algal proteins and starch, characterize the physicochemical properties of the extracted algal polymers, biopolymers, and develop algal protein-based seafood analogs. Let's look at some results. First, the protein extraction and characterization. So the ulva has about 9% protein and Gracilaria about 25%. And to extract proteins from seaweeds, basically there are several steps. First of all, cell lysis. That could be done by a variety of methods like osmotic shock, sonication, bead milling, pulsed electric fields, so on. The next step is the extraction, and here usually alkali solvents are used, or reducing agents or salting out agents to get the proteins out, particularly one stuck in the membrane. And finally, separation and purification, like using dialysis, centrifugation, ultrafiltration, and so on. So we picked sonication, alkali solvent, and dialysis as the main steps. And we managed to come up with a food-grade protocol. We compared it to four other protocols found in the literature, which are not food-grade. And we managed to get better results in terms of the amounts and purity of the proteins that we extracted. We got 62% protein. Most of the proteins, as you can see, are between 5 and 15 kilodaltons with some higher fractions, higher molecular weight fractions. We then tried to get even higher purity by using ion exchange. This way, we managed to get as high as 86% from the gracilaria, about 71% of protein in the extract from ulva. The amino acids composition was very interesting. We have good, high quality amino acids like high concentration of lysine, both ulva and gracilaria. You can compare it to ovalbumin from eggs, but also compare it to soybean and zein, so it's much higher than the plant. Also, threonine, valine, leucine, quite high, better than some of the plant alternatives. Also, in terms of protein digestibility, we did simulated digestion and compared it to casein, and we found that gracilaria was even more digestible than casein, and ulva was slightly less, but quite good. And you can see the diminishing of the proteins during digestion, the gastric phase, the intestinal phase. And another interesting property was the antioxidant activity. So using the ORAC method, we compared to 3 different proteins like serum albumin, beta-lactoglobulin, and potato protein, and we found ulva protein was similar, and the gracilaria was much higher. And interestingly, during digestion, the antioxidant capacity even increases significantly due to the exposure of various groups and breakdown of the protein. And so to conclude the algal protein part, developed a new food grade protocol for extraction. And we got extract with over 70% protein, including all the essential amino acids, high digestibility, good antioxidant activity. And what about starch? So first we wanted to find out, and this was mainly done by the group of Alvaro Israel, Professor Alvaro Israel in the IOLR. And they looked at the conditions. Is it good? Is it preferable to give fertilizer when you want to produce starch in ulva? Apparently not. If you want to get high amount of starch, you prefer not to put fertilizers, because when you put fertilizers, you get a lot of growth rate, a lot of protein production, but not a lot of starch. So if you want to then get a lot of starch, you have to do some nutrient starvation, and then they start producing starch as a backup energy source for bad days. And so you can play with that if you want to produce starch or protein. The process of extracting the starch was mainly developed by the group of Alexander Goldberg, and they took the raw biomass, scrubbed the cells, filtered them, centrifuge them. They used ethanolic wash to get rid of the green of the chlorophyll, and finally they got starch granules. Interestingly, the size of the granules was much smaller than, for example, potato starch, and they're about 7 micron compared to 48 micron of the potato starch. And what is even more interesting is the amylose content. In most plant sources, amylose content is relatively low. For example, in rice starch, it's 34% of the starch, and in potatoes, it's only 24%, while in ulva, we found 55% of the starch was amylose. That is very interesting and very useful because amylose helps gelation, and helps also retrogradation. And as in a minute, I'll show you implications. For example, we looked at the biological properties when you do a gelatinization of starch. So here we looked at the viscosity, the time of heating. So this is the heating as process. First we heat it, then we hold it, and then we cool it. So you can see the increase in viscosity upon gelatinization. Then during holding and stirring, it goes down. And during cooling, you build a gel and it rises again. So we can look at peak viscosity and breakdown viscosity. This is called the trough viscosity, and finally the final viscosity. And if you look at the ratio of the final viscosity to the trough viscosity, that's a good indication of the retrogradation. The retrogradation is a process where starch starts to crystallize, to recrystallize, and those crystalline regions are like crosslinks, which help build the gel. There are also areas which are much less accessible to digestible enzymes. So on the one hand, we get higher gelling, which is good for functional properties, but on the other hand, we get less digestibility, which is good for health reasons. We get less digestibility, so lower glycemic index potential and so on. We also looked at the molecular weight of the starches, and you can see Urva had a relatively low molecular weight, slightly higher, and potato higher. And we looked at the crystallinity. So typically, the most crystalline is the initial granular stage. Then during gelatinization, upon heating in water, we lose all crystallinity. And upon cooling and retrogradation, we get some crystallinity reforming. That's the retrogradation. And so during digestion, our body breaks down the starch with the various amylases. And as you can see, the least digestible, the least bioavailable and digestible is the native starch, granular starch. Most bioavailable and most digestible is the gelatinized starch. And the retrograded starch is less digestible. With the uva starch, least digestible of those three types of starches that we looked at. And this suggests that potentially the glycemic index would be low or lowest, which is a good news. And so overall, we have good rheological properties, good more rigid gel structure, more crystallinity, which means moderate hydrolysis, less accessibility to enzymatic hydrolysis. And so lower digestibility means lower glycemic index and even lower caloric value in potential. So these are all good news. So to summarize, size, smaller granules, small molecular weight, compared to the terrestrial plants. Amylose content was relatively high compared to terrestrial plants. Good rheology, higher, stiffer, firmer gels, and digestibility is lower, which is actually good. And lastly, we're now working on trying to use extracts of the seaweeds to create plant-based seafood meat analogs. So basically, if we take plants or algal-based raw material, we want to extract the protein and use various structuring and formation, texturization, and formation technologies to form seafood analogs. Okay, so let's see some results. We decided that get the texture and the structure of the seafood, maybe it's better not to extract the protein too purely, because we would like also to make use of the gelling properties of the polysaccharides. And also in terms of minimal processing, we want less processing, so it's more efficient this way. And so we kept much of the polysaccharide fraction, and the techniques we used are scalable, and we extracted an extract which contains about 50% protein and some and a large amount of polysaccharides as well. Interestingly, the proteins have an electric pH of around 5. So if we go below 5, we have opposite charges with slightly negatively charged agar. And so at high pH, we see two different peaks when they're repulsed. And when we go to lower pH, when they have opposite charges, we see complexes with one peak, and the zeta potential of the complexes was like that, decreases with pH. And now we're working on the final stage of 3D printing. We want to take those concentrates and try to print them into a fish steak or fish fillet. And so we got a model of a fish fillet, and we are now working on its 3D printing into something which would be eventually edible. And so that's the vision, still in the process. So to conclude, we want to retain the polysaccharide fraction, some of it at least, characterize the electrostatic attraction with the 3D printing, and hopefully in the next steps, we will look also into texture, taste, and optimize the process. So overall, our study promotes the use of macroalgae as new and renewable sources of food ingredients, like in particular proteins and starch. This is our team, and this is Meital, who did much of the work you've seen. And I would like also to acknowledge the Good Food Institute, the Ministry of Science and Technology, and the Ministry of Health for funding these projects, and for you. Thank you for your attention.
So, typing presentation. So we have 2 minutes for questions. If anyone has a question, Sami, Professor Sami Wasiba.
Well, I was impressed by your downstream processing. But the problem is not downstream processing. The major problem is the production of this biomass, where and how. And we are so far from trying to reach the substitution of soybean. You know how much soybean is produced? 300 million tons. You know how much it costs? $0.50 per kilo protein. So how we, with this kind of solution, can help saving the problem. It is okay if you want to make a boutique production, if you want to sell in the supersal or market some fish or you know, made from algae. This is okay, I agree, you can make money, but we're not going to solve the problem.
Well, of course, you're raising very important points. I will try to address it. It's not easy, but I think there are 2 things. First of all, there are larger areas of water on Earth than land. And we may be able in the future to develop offshore farms to grow seaweeds in large amounts. Area is not the problem, but I believe that with time, there will be more and more problems in growing the current crops because of global warming and growing world population. So prices of the land-grown plants will rise. And so at some point, probably in the future, if we manage to develop efficient offshore production for algae, there might be a time in the, I don't know, 20, 30, 50 years from now, where the prices will be competitive. I'm sure it's not in the near future, but we need to look into the future and start working on solutions for bad days ahead.
Yeah, but the best solution, to my opinion, is to try to do things that we do now much better, more efficient, so on and so forth. All of the other stories, I call them a boutique production. It will be boutique production, but it's not something that will solve. But let's see what the future will say.
Exactly. Yes. With these nice words, I would like to thank Professor.
So the next speaker is Orr Shapiro from the Khan Institute. He will talk about with machine learning algorithms for monitoring the quality and safety of algal biomass.
Okay, so first of all, thanks to Ina and Dina for inviting me here. A great conference. And I changed a bit my title because after I submitted my abstract, I learned that Olubunmi, my student who did most of the work I'm going to present here, will be talking after me. So it's kind of a prelude to her talk as well. So general spectral methods for management and monitoring of microalgal bioreactors. So we're going back to micro. I'm going to skip over that because I was asked to be short. So yes, we know that microalgae have a lot of potential. Some species are very rich in protein or in lipids, have a good potential food source. And we already heard about the different systems for growing these kind of microalgae. And also we already had
by the way, on the left side is lemna, it's not algae. So correct your slide next time.
Okay. Excellent. I think the third one is spirulina, but I might be wrong.
No, it is lemna.
Okay.
Believe me, it is lemna.
I'm corrected. Okay, but it's similar to spirulina. I can argue that. Anyway, we know that these kind of systems are widely used in many microalgae, but we already heard the term boutique product. So these are relatively expensive and take a lot of effort to manage, which means they're not the solution for producing food or definitely feed within affordable prices. And then we go to these kind of raceways. So this might be lemna, but many spirulina cultures are grown in this way as well. And either open or in some kind of a greenhouse, which is a bit more closed. In either way, we have a lot of issues with these kind of systems in terms of quality control, in terms of culture crash, which we already heard about today. And specifically today, I'm going to talk about contamination events, which are a major problem, especially in these kind of open systems, even though these systems too get contaminated all the time. So it's true for all of them. And this is actually, this paper came out about the time I started working in Vulcani, and was sort of a trigger for a lot of the work that you're seeing here. And it's a work from Greece that took 31 spirulina products from the shelf in the supermarket and just tested what other bacteria, because spirulina, of course, is not an algae, it's a bacterium, it's a photosynthetic bacterium. What other bacteria are in these products? These are finalized products that are sold in the supermarket. And they found that 54 first of course, bacteria are found in all spirulina products, including stuff that you don't want, like Pseudomonas and Vibrio cholera. But we can assume that they're not pathogenic, or at least not active after the downstream processing. But 54% of the products they analyzed contained additional cyanobacteria species. So that's not very surprising, because if you grow spirulina in an open environment, you give all the conditions right for growing cyanobacteria. It's not surprising that cyanobacteria grow there. So other cyanobacteria tend to invade the system and proliferate. Specifically, one product had almost 50% spirulina. The rest was mostly Mycocystis. Mycocystis is a toxic cyanobacterium that is a pest in a lot of freshwater systems, including the green water aquaculture. It's a huge problem there. We have other projects on this in the lab on these kind of problems. But that's, of course, not an isolated report. There are many other similar reports from China, from the US, from finding similar level of contamination. In China, all 36 products were contaminated with mycocystins. Here as well, this is also in Greece. Again, all 31 products tested positive for mycocystins, for the toxin, for the Mycocystis. And another one here, finding 40 or 51 products tested positive again for mycocystins. I should say that most of them are below the toxic threshold as defined by the FAO. but it does mean that most of them are contaminated, and potentially they can get to toxic level. Here, 8 products were above the toxic level for adults. Here, several were above the toxic level for children. So it's a very, very common problem in these kind of systems. Now, what can we do about it? What is done today? Mostly when you talk to growers, they tell you, oh, yes, we do daily microscopy. Take samples to the microscope, and we look to see if there are any contaminants. And I'll show you later, this is not the best approach, but this is the state of the art. You can do pigment analysis if you're growing microalgae, not cyanobacterium, then you can just use pigment fluorescence to differentiate, but it's not always that easy or that precise. And of course, when you have already cyanobacteria in the pond, you cannot detect other cyanobacteria. In the lab, we can do other very precise things like real-time PCR, we can do ELISA, we can do different types of chromatographies, but these are not practical in the field when you're doing agriculture. This is not going to work. We're looking for other methods. And one very tempting way to go is using different types of spectral methods. So what are spectral methods? When you shine a light on an algal culture, you have different types of interactions. So first of all, of course, how the light gets absorbed because they do photosynthesis and molecules absorb light, so that's obvious. Then you have some light that is reflected, and this is basically the most basic type of spectral analysis that you do is just looking at the culture. And a good grower will be able to say, well, the color is not right, something is wrong with the system. But this is a bit too late if you get to this stage, and definitely not very precise. Then you have the transmitted light. Olubunmi will be talking about this kind of system later. We have fluorescence. We mentioned pigment fluorescence and other type of fluorescence in the system. But what we started working with, and this was Olubunmi's master's and later on part of a PhD, was Raman scattering, which was a system that was used in the engineering department for post-harvest analysis of fruits and vegetables. Actually. A very crude system where you go and you take a Raman system, you touch fruit, and you analyze sugars or lipids. So this kind of post-harvest approach. I'll talk about it a bit later, how we do it. So what is Raman scatter? Just very shortly, when we have light hitting a molecule, we get some kind of vibration with a very specific frequency of the molecular bond between the two atoms. And the specific vibration depends on the type of atoms in the bond and also on neighboring bonds in the molecule. And then you get what is called Raman scatter, which is light and the energy that is proportional to the original frequency of the light, plus or minus the vibration of the molecular bond. We'll ignore these parts right now, and we're only talking about the light that has lower frequency, so longer wavelength that is emitted from the sample. I should say that this is a very, very rare event, these kind of systems, so you have very sensitive spectrometers to detect Raman scattering, but it is practical. And this is, for example, the Raman spectrum of ethanol, a relatively simple molecular molecule. And you can see that you have these specific peaks that are very, very characteristic of each type of bond within the system. Of course, what happens when you go to a biological system? Most of the time when you hear Raman of microalgae, you see this kind of analysis of Raman microscopy. And here they're looking at Desmodesmos, and they're looking at different areas within cell, and you can see that where do you have crystalline guanine, you have some very clear peaks, but in other places where they have phosphates or starch, or just what they call other biomolecules, which are probably lipids, proteins, DNA, what have you, you have something that is much less clear and robust, and also very, very similar, of course, to each other. So you can see this peak and say this is probably the starch, and this peak here might be representative of the polyphosphates. So of course, it's a much less clear-cut story, and it's harder to analyze. And we're going even a step further, because we're saying we don't want to look at one cell at a time. That's not practical in a commercial system. We want to work like we're working with the fruits and vegetables. We want to look at the bulk culture. We took the system using the agriculture engineering and we diverted it to something that can work with the water samples. Basically, we have the laser source. We have a shutter here to reduce fluorescence over time and the changes in the fluorescence of the cells. We have a sample chamber which holds basically a PCR tube with 300 microliters of the raw culture of the algae. So we are directly measuring the culture within the algae. We know pre-processing needed here. This goes to the spectrometer and to the computer. And within about 8 seconds, we can get a reading from each sample in this case. I should mention here Dr. Timea Ignat, which helped us a lot with setting up the system and then moved here. Now moved on to industry, unfortunately, but it was a great help in setting up this system. So this is what it looks like when you start taking measurements. Some of the first measurements that Olubunmi took with spirulina, because we started working on a model system of contamination in spirulina. And you can see here, these are spectra from cultures from 10 to the second, 10 to the third down here, all the way to 10 to the 8 cells per mil up here. And two things are immediately clear. First of all, 10 to the second, 10 to the third are very close to each other. Might not be very easy to tell the difference, even though there are differences here. Then this is 10 to the 4, 10 to the 5, and already 10 to the 5, slightly above that, we're starting to see this hill and the peaks get very, very low. And this is because of fluorescence of the cells. This can be overcome, but not with the system that we have here. It will be future generation of the system. But we worked with this because we said, yes, but still the fluorescence is part of the characteristics of the system, even though it's not true Raman, can still work with this. This is where the magic happened, and I'm not going to go into the machine learning algorithm. You can talk to Olubunmi all you want about that later on. But you have to extract information, you do pre-processing and design the chemometric models and test and validate to make sure you're getting something that you actually believe. It's always dangerous with the machine learning to believe what you get. And finally, we can do quantitative and qualitative or classification models to actually see what we can tell about our cultures based on this kind of spectra. First of all, this is what the different spectra look like, and you can see some similarities, some differences. First of all, there is always this kind of hill around here, and it's more pronounced in the cyanobacteria than in the microalgae, but it's always there almost. We wanted to see the sensitivity, so we're just doing here a correlation of the concentration of the cells to the readings and trying just to infer concentration. And we can see that we can go with a decent linear fit all the way down to 10 to the third, sometimes even 10 to the second, but getting hard at these areas. But definitely from 10 to the third to 10 to the 8 or 10 to the 7 for some species, we can get a very good fit, which means we're at least sensitive to this range of concentrations. Next, we just did a very simple, this kind of like a PC plot, PC analysis. And you can see that just based on the spectra we got, by the way, this is all of the different concentrations that we took from 10 to the third to 10 to the 8. And already we can see that we can cluster them just based on the spectra, which means that even though to us it's not always immediately evident, there are differences here that this kind of analysis can detect. So can we actually use this for analyzing the samples? We started again simply, we just think, can we tell the difference? This is just the question of these 10 to the 6 cells per milliliter cultures. Can we tell the difference between a spirulina sample here and all the other ones? Obviously we can, and this is microcystin. We know that this contaminant that happens in a lot of spirulina system. And again, we can tell the difference between spirulina and all the other cyanobacterial and algal cultures that we have here. Sorry, I don't have the names here, but it's basically a random collection of algae and cyanobacteria that we had in the lab coming from fish ponds that we were working on at the time. We could have made better choices, but it was useful. Then we're going down to 10 to the 4 cell per mil, and we're still doing a pretty decent job at separating spirulina from all the other ones. And again, microcystis, we have some glitches here, but overall, we're doing a good job of 10 to the 4 cells per mil. Any of you who held the 10 to the 4 cells per mil culture, it's basically water, right? It's transparent. You can still tell the difference just with 8 seconds of measurement. So it's working. This is a confusion matrix, just comparing all of the different species to each other, and we can see that with 10 to the 6 cells per mil, we can do an overall accuracy of 95%, meaning 95% of the time I can tell you what sample I'm holding in my hand just based on this 8 seconds of sampling. And then this 10 to the 4, we are still doing 85%, which is pretty decent. So this was very encouraging. And here we're going on to the question of, can we detect contamination events? Contamination events, of course, start very low. You start with very low levels of contamination, it grows. So we're starting here with a model culture of 10 to the 8 cells per mil of spirulina. And we're adding contaminants, in this case, the mycocystis and Myosmopedia and the Desmodesmus. So this is the Nadreciano and this is the microalgae. And we're adding contaminant with 10 to the second to 10 to the 8 cells per mil. And I must say we expected a lot more spread here, but very surprisingly, we were asking, we built a model that was asking, is this clean spirulina? And for the spirulina, most of the time, the answer was yes, this is clean spirulina. This was the answer for all of the contaminated samples, including the ones with the 2 second cells per mil. This was almost unbelievable. I didn't believe it. We had to repeat, but this was the result. So somehow with the Raman, we have the ability to say this is no longer clean spirulina. And I can't tell you what we're seeing, unfortunately. We haven't yet got to the point where we know what we're looking at, what the system is seeing, but it is able to detect the contamination. And here I should go back to the point that usually you're using microscopy. Now with microscopy, you remember that culture that had 50% spirulina, 50% mycocystis. This is what it would look like under the microscope. So somebody did a very bad job of monitoring the system there because it's very obvious. When you go down to 10 to the 7 cells per mil of mycocystis, you can still see them, but obviously it's not as clear. In 10 to the 6, in this field of view, we have 3 cells. That's it. And this is 1 in 100 cells of a toxic cyanobacterium. With 10 to the 5, if you have 10 to the 5 cells per mil in a culture of 10 to the 8, you won't see them. You're not going to see these cells. And if you concentrate, of course, you get blocked with all the spirulina. With the microscopy, you will not be able to see 1 in 1,000 cells of mycocystis. 1 minute. Okay. So this is definitely doing a better job. Now, who is there? Can we detect, can we identify the contaminant? When we work with 10 to the second, 10 to the third cells per mil, contaminating with mycocystis aeruginosa, we can tell most of the time that yes, this is the contaminant. We're going up to 10 to the 4, 10 to the 5 cells per mil, 100%. And of course, we get 10 to the 78, it's easy. So we're doing a pretty good job here at detecting and identifying the contaminating species. That's important because you want to know if it's a toxic or non-toxic. Who is the, these are the other 2 species that we're testing. And again, very similar results. We have some misidentifications here, especially for the lower concentrations. As I said, we can at least say that it is contaminated, even if we still can't say who it is. How bad is it? How many do we have? So this is, we haven't got to the point where we have the specific, the quantitative model, but just from, again, the PCA analysis, you can see that we are doing a good job of differentiating spirulina from the higher contamination. All the lower ones are grouped together here. But if we zoom in, we can actually tell the difference, which means the information is there. We still have to extract it. But we should be able to tell the concentration of the contaminant within this kind of a system. I should say that these are all very, very clean cultures in laboratory settings, growing under very, very stable conditions. So this is not real life. Olubunmi is now doing something that is a bit more like real life, growing your own cultures in bioreactors, contaminating them, and trying to see how this works. We don't have the data for that yet, but it's coming. And so conclusions, yes, I think this kind of low resolution Raman has the ability to detect this kind of contamination events very early on, and possibly other problems, like if you have some problem with your lipids, with your pigments, whatever it is you want to produce in your system, the Raman should be able to see it because it sees molecules. So it's only a matter of having enough background information of what it needs to, what it should look like, and then you should be able to say when it's not looking right. So it's all about training the machine. So these kind of approaches should in the future give us a better tool for very quickly, very efficiently tracking, monitoring the state of our systems and raising a red flag whenever we have a problem. And just thanking the team in the lab, specifically Olubunmi, who is going to talk in a couple of talks. Gigi, who is going the algae, Shmilo and Timea, and Itai, who is collaborating on the HTS system. We'll see you later, and thank you.
Half a minute for a very quick question.
So if you have a contamination of 0.5 algae in spirulina, you throw the spirulina 4,000 liters?
That's a good question, and that's I'm not going to answer. So it's up to you, okay? Because you're going.
No, I mean, if it is high, you'll see it on the microscope. So
Yes.
I don't know.
But no, I know. I anticipated the question because you're saying, okay, now I know what am I going to do about it? And the answer is, I don't know. Sometimes maybe you can do something about it. Definitely with spirulina, you can try pushing the pH higher. This might subdue the contamination, depending on the contamination. At least it might tell you that this is, yes, you have a contamination, but it's not going to be a problem because it's not toxic. It's not toxic. Already you're selling stuff that is not useful. So again, one thing that I haven't mentioned here, we don't have regulations today. One day you're going to have the regulation. When you're going to have regulation, what are you going to do about it? Because if you're going to use spirulina to feed fish to produce salmon-like substances, we have a group in Israel doing that, to use this for nutrients for future meat. Also,
don't frighten us, because spirulina has been produced for the last 40 years, and until now, I never heard something bad about it. So please, hold your horses.
Okay.
Okay, after the session, we still have some time. Thank you. So we'll now move to a lecture of early career scientists or early career researchers. You have a double challenge because you have only 12 minutes per lecture. So the first is Dr. Avraham Ben-Sheleg, a student at our institute. Let you introduce your topic.
Dina, and thank you for the opportunity to speak here. So, ROS is produced by the photosynthetic apparatus, and that is used due to lack of efficiency along the photosynthetic apparatus, meaning that ROS is meaning the energetic load on the photosynthetic apparatus along where the energy is lost along the photosynthetic apparatus will tell us what type and how much of ROS is produced. So singlet oxygen, unlike other forms of reactive oxygen species, is unique in its creation. Other forms, we generally talk about transfer of an electron from the photosynthetic apparatus to oxygen. Well, singlet oxygen is created due to the interaction of oxygen with a photosynthesizer. When we're talking about endogenous production inside the photosynthetic apparatus, usually we're talking about triplet chlorophyll as our photosynthesizer production of ROS, of singlet oxygen. So, in other words, we have is a unique sort of singlet oxygen has become unique in the fact that it is its production site is almost exclusively inside the thyroid membrane, and specifically, we oftentimes associate it with photosynthesis. So, which is also why a lot of signaling pathways, especially in plants, have been discovered, right? Because we have this very specific molecule associated with the energetic load on our light harvesting complexes. Many signaling pathways have been discovered in plants. Again, it's been always when we're talking about the different pathways of singlet oxygen signaling, we're talking about how much singlet oxygen is produced and is relative to the energetic load in our system and which molecules it is interacting with carcinoids. And for plants, well, there have been several molecules discovered that interaction can trigger a signaling pathway. All those molecules have been found in the thylakoid membrane, in the lipid phase. So in green algae, it was explored whether a signaling pathway exists. They found that light can induce a photooxidative response. Similarly, Ross Bengal, which is a photosynthesizing dye that can produce synthetic oxygen, also could produce a similar photooxidative response. Expression of GPA. What was seen in the light, but it did in a dark environment, showing that response, evidence that response happens due to oxidative. Interestingly, when they inhibited carotenoid and other antioxidant synthesis, which have been found to be significant players in oxygen signaling for plants, though that had no influence on the response of the green algae to single oxygen or light response. Furthermore, they found single oxygen in the aqueous phase was detected. So together, they put together a model that for green algae could have a pathway that may exist outside of the thylakoid membrane. Then further prove this in the PNS. So, but the question is, all this sort of research has been done until now is focused sort of on one lineage, right? Green algae and plant lineage. Because we all know, and which was discussed by people before me, that algae is much more diverse than that. And there are many different lineages, many of which have their own significance biologically or biotechnologically. We focus on the heterocon, well, specifically the model species Nanoclotha oceanica, which to as its model as fatty acid synthesis species. And so this is both a very genetically diverse and morphologically diverse due to its heritage. I'm not going to get into it due to time limits. And the question we want to ask is, what happens here? Is there even a signaling pathway? And how does signaloxin also affect damaging the photosynthetic apparatus? So we created a system where you can see in green, it's supposed to be, green is the growth of anacharopsis in a Ross Bengal environment, which is actually a photosynthesizer. And this is the control, which is just growth in a low light, same low light environment. And what we found, then we wanted to check various photosynthetic parameters, see what it was like. And we compared where we can see blue is our reference, our low light reference, relative to our green, which is our same environment, just with our Ross Bengal photosynthesizer in it. Red was a reference growth in a highlight environment. First thing we could see is we look at our parameters such as FV life, and all these are normalized to the parameter value inside the low light. You can see that, first of all, we can look at some parameters that have to do with stress, such as FV or FM. We saw no damage which occurred, even though there was a complete inhibition of growth or death inside our Ross Bengal culture. Indicating to us that the single oxygen had no, at least our exogenous production sites had no effect, no damaging effect to our photosynthetic apparatus. But all the other parameters, such as our maximum oxygen productions, our photosynthetic efficiency indexes, even our diffusion of energy around our photosynthetic sphere, which is oftentimes correlated to loss of energy around photosynthetic, which is oftentimes correlated to the production. The oxygen himself behaves similar to a highlight. Okay? So meaning we saw very strong evidence to highlight acclimation. It isn't surprising because single oxygen has been already pathways, but we saw this without any damage to photosynthetic apparatus, indicating that Ross Bengal acted presumably outside of the chloroplast and still triggered this sort of response. I actually wanted to look at this from the opposite angle. Okay, so we know that Ross Bengal can target some molecule outside, likely outside of the chloroplast, plastid. And that molecule will trigger some sort of acclimative response, highlight acclimative response. The question is that molecule also significant to some sort of to functioning of photosynthesis in a highlight environment. So Noritus here did mutagenesis of this nanocalypsis. The EMS caused the mutagenesis, and Ross Bengal as a selection, as our selection plates. And what we found is the mutants who were grown, isolated due to their resilience to Ross Bengal, grew better in highlight environments, meaning a highlight environment which lethal to our wild type, was not lethal to the Ross Bengal. And furthermore, this death of the wild type, which was correlated to lipid peroxidation, loss production, no such loss production was found inside. Furthermore, we saw that along with the production of ROS, we saw destruction or damage to our posting apparatus into fluorescence data. And what we saw was the productivity of our mutant lines increased, not because we saw any serious changes in fatty acid production, but rather just because fatty acid production was maintained in the biomass significantly inside our mutant string. This was all. This I'd like to thank all the other players in this project. I don't know my libraries. Okay.
Thank you, Boone. We have time for questions. One short question.
It's nice approach, like to simulate oxidative stress by Ross Bengal, but like, do you have impression like after you work that like you can simulate the oxidative stress like in algae? Maybe it's like with some another like agents, it would be like more similar actually to the real like the light damage agent, like to the sarcoid membranes and to chlorophyll. Like you apply Ross Bengal and you get some results.
I use also metivalogen, which did damage. I use other. I didn't show all the data, but yeah, we did use other. I had to create one story here, but yeah.
Did I understand correctly that you were saying that Ross Bengal does not damage something in the code?
We didn't find evidence that did, right?
Very surprising. Can you explain why? Because Ross Bengal is a generator of singlet oxygen,
Right.
which is primary targets at polyunsaturated acids of chloroplast membrane, the state of the art.
Polyunsaturated fatty acids. So it probably does target polyunsaturated fatty acids. But we also have to remember that in nanocalypsis, you have more layers of membrane. It has so I can speak about, I'll answer you in two, right? I'll tell you what we do know, I'll tell you what we don't know. First of all, there was a PNAS, right, that I didn't show here, but that answered that they found in Chlamydomonas, really, that also reactive, the interaction with any sort of, lipid peroxidation occurs, any peroxidation at any membrane can actually trigger the same sort of oxidative response. Imagine whether or not it was produced inside the chloroplast or was produced somewhere in the vacuole, other organ. That was shown in Chlamydomonas that interactions with lipids, not only inside the. So likely it's something similar here. Also, when we did do DNA, we did genome analysis, and we actually found an interaction with.
Thank you. So our next presenter, Miss Olubunmi Adejimi, thank you, of the Hebrew University and also the Volcani Institute.
So thank you for the opportunity to speak here. It's an honor for me. I'm going to be talking about using hyperspectral transmitters characterizing microalgal cultures, supervised by Dr. Herman at the Faculty of Agriculture in Rehovot, and who you heard about earlier, I heard from. Okay. So I'm not going to be covering a lot of this since we already got but mostly it's just in this case, we're going to be looking more about the potential for using this method, this spectroscopic method for pigment monitoring, and then we would also be looking at the possibility of also using it for contamination monitoring. And for us, anyhow, the basis of the research is just to find methods that are sensitive, affordable, fast, and easy for growers to monitor their microalgal bioreactors. We also got introduced to this. So Orr talked about the Raman scatter, but I will be talking about the transmitted light through the cultures, which pretty much. So one of the major the highlights of hyperspectral transmitted spectroscopy is that it's has high resolution. So you have 2,150 variables or wavelengths in this case. And this is, I think the best way to think about it is to think about the OD, the commonly used OD by biologists, and then you're taking this OD information simultaneously. So you're not scanning, it's a simultaneous measurement. And a lot of the information is for pigment and very useful for bulk analysis, very fast, very easy to use. And we already heard about the Raman spectroscopy, which is very highly sensitive, especially to molecular. So setup is also quite simple. This is also, like I said, easy to use. We want this method that is very simple, and you have like the light source and the light passing through the culture, and we collect the transmitted light information displayed on the computer. And what we do is that we combine this with the chemometric methods, or we explore the use of chemometric methods, and also combining it with the machine learning algorithms for quantification and for classification. So this is what the spectra looks like. So as we see, we have this region here that is we have like very fingerprint-like spectra or information. But then beyond that, we have these areas that are much less informative, and this region that is just very noisy due to environment, not necessarily environment, just a lot of noise. And what we decided to do was, okay, what part gives us the best information? And we saw that it was this region over here, which is between 400 nanometers and 750 nanometers, where we decided to focus on. And this was the information that we used for the further analysis that we did. So the first thing was to see, okay, can we still do OD even with this information, or how can we use the OD information that is already present in this hyperspectral spectrum and use this for quantifying our cultures. And what we did was to take at 440 nanometers and 680 nanometers, which are representative of the chlorophyll A pigment, present in every micro or photosynthesizing species. And what we saw was that using this information, we couldn't get a very clear linear correlation between the concentration and the transmittance at these wavelengths. So what we did was to create a threshold, and what we noticed was that at the lower concentration, we were not doing very well. We had very poor R-square values, and we had also very poor RMSE values. But at the higher concentrations, so that we had very high R-square values and very low error rates. And this just tells us, and we observed this both for spirulina and for desmos, and even for all the other species that we looked at. I'm not showing everything here just to save time. And the conclusion we came to here is that OD is great, and but it's pretty much very useful only at higher concentrations. At lower concentrations, you cannot be sure of whatever predictions they are getting in terms of quantifying the cultures. So what we did next was to now take the hyperspectral measurements, the full hyperspectral measurements with the machine learning algorithm support vector machine. And we decided to see, can we actually make this quantification? And here we can see that we get a more linear relationship between the measured concentration and the predicted concentration from the algorithms. Here we have a bit more of a variation in the in predictions, but this is also because the spirulina cultures that we have in the lab are not exactly homogenized. So we have I'm sorry, I keep forgetting words, but we have clumps within the culture. So this is why we have more of an error in this case. But with the more homogenized cultures, we're able to get very impressive R-square values on RMSE. And it's important to note that these results that we're looking at here are from the validation set of the data. So this is not the calibration. So, and when we compared, just to compare the metrics for the ODs and for the machine learning methods, we see that the machine learning is doing significantly better than the OD for the R-square, and even have generally lower RMSE with the machine learning algorithm. So we know that if you want to find a method that covers every concentration range that you will be looking at, then using the full spectra for quantifying, and the machine learning algorithm would be the way. And so the next thing for us was, can we classify? Because we can quantify all we want, but I think a lot of people will be more interested in knowing what are we actually looking at. And we decided the first step we did was to use the full resolution of the spectra. And we just calibrated the model. And now I'm showing the results for the validation set. And we see that at the lowest concentration, which is 10 to the 5th cells per mil that we're working with here, we were not able to predict the spirulina samples at that concentration. But once we go to 10 to the 6 cells per mil, we start to see, it's important to know that the y-axis is the probability for all these samples to be the mentioned species. And the threshold just says it's a 0.5 threshold, which is just the equidistant point between the highest probability and the lowest probability. And so we see that already here that we're able to predict at 10 to the 6 cells per mil, and also at 10 to the 7 cells per mil, we're able to make correct predictions completely for the presence of spirulina. And we see a similar trend also with desmosdes. And I want to say again that it's the same story with all the other species that we looked at, the other cultures that we looked at. And important to know, this is pretty much what a 10 to the 6 like. So we're even able, knowing that the microspectral transmitting spectroscopy is less sensitive than the Raman, we're still able to predict even at cases at 10 to the 5 cells per mil for this species, maybe not with the spirulina. So with this information, we decided to look into, okay, what parts of the spectra, what wavelengths are actually important for making these kinds of predictions. This is what we found. We used there are two ensemble models to do this. And what we found was that these regions here, which are predominantly like the chlorophyll region, especially this area here, we were able to get the peaks that were most important. But how we made our threshold was to ask if all the wavelengths had equal weights or equal importances, what would be the value, and then we picked every wavelength that had a weight above that value. And what we found was that with a reduced number of wavelengths or reduced dimensions, we're able to even improve our predictions compared to what we had with the full resolution. And we did the same for spirulina and for desmosdes, and this was what we observed. And I think this for us indicated that we do not need the high dimensionality of the hyperspectral data. Well, it's good and it's information rich, but maybe we can actually still make the predictions we want to make with lower dimensions or lower information. So what we decided to do next was to explore a method called feature aggregation. And this is just aggregating the neighboring wavelengths with n width. And anyhow, this is what we did. We made aggregates along these lines, and that was what we did for the whole spectrum. And when we tried with n values between 1 and 50, we saw that using an aggregate of 10 wavelengths gave us the best accuracy, and see the result very clearly here, where We can clearly see that we're doing better, even with the spirulina. Remember, we couldn't actually make predict the lowest concentrations for the spirulina. And we're doing the same also with the desmodesmos much better, even though we have some false positives here, but this is, these are already species that are quite similar to each other. This is important to remember. So we're comfortable with that for now. We can improve, but for now, we're comfortable with the results that we have. And when we compare between the full resolution, the feature selection, and the feature aggregation, we see that feature aggregation gave us the best result. And we think that this is because while we're aggregating the features, we're still including some of the information from the neighboring wavelengths. And this allows us to be able to include that information within the machine learning algorithm and to be able to make better predictions. The next thing we wanted to do was just to see, is there even a possibility for detecting contaminants? And here we see that, okay, that's fine. We see that we can only detect at the highest concentration of contamination by Microcystis aeruginosa. And, but with the PCA, we see that there's still some of that information. As you can see at PC3 and PC4, there's still some separation. So we know the information is there. We need to dig in, find the right algorithms to use this information. So in conclusion, we've been able to show that we can quantify microalgal species. We can apply, only use simple chemometric methods for high density cultures. And we know already, we can see that high resolution transmittance spectra does not, we don't necessarily need the full resolution, the high resolution. We can do with a reduced resolution of the spectra and still be able to make very good and very impressive predictions. And we see that there are a few applications we think that we can use this for monitoring pigments and also for the monitoring of starter cultures for eventual use in bioreactors. And with that, I say thank you to the members of the both labs that I'm affiliated with and to all the sponsors and to the DDD conference. Thank you very much.
We don't have time for questions, so after the lecture.
Thank you. Yes, I would like to tell you and to Orr if you would like to discuss after, if you have time. Okay, who is the next speaker? He will talk here about phosphorus uptake and what he is an expert in remote sensing and Raman and spectroscopy, non-destructive, optical spectroscopy of microalgae. And so by this, I welcome Alexei to the stage. Alexei is a professor at Moscow State University, and he will talk about biocapture phosphorus. It's a very emerging and important topic.
Thank you, Ina. Thank you. So, phosphorus. Why at all to talk about phosphorus? Listen to the talks, this conference, and why on the tour yesterday, I was hearing a lot about nitrogen use efficiency, and it seems like everybody concerned about the lack or shortage of nitrogen. And almost no one thinks about the deficiency or shortage of phosphorus. But phosphorus is also central to the metabolism of every cell. It's central to the exchange of energy and information, absolutely essential and cannot be replaced by any other element. Phosphorus is called the element with a broken geochemical cycle because it does not have volatile compounds which can close the cycle. For example, for carbon you have CO2, for oxygen you have dioxygen, for nitrogen you have N2. That's not the case for phosphorus. Moreover, phosphorus is a lifeblood of modern agriculture. That if you don't have phosphorus fertilizers, you don't have high crops. And phosphorus fertilizers are made from rock phosphate, non-renewable and very unevenly distributed resource. By different forecasts, it's expected to be finished by 2015 or even earlier. As paradoxically as it can sound, current phosphorus supply chain is very inefficient. For nitrogen, we have the efficiency of about 30%. For phosphorus, even less, something like 20%, meaning that 80% of what we mine from rock phosphate mines ends up in waste. And this is not only wasteful, this is also dangerous to the environment, because lost phosphorus ends up in water bodies, and it causes eutrophication and all the negative consequences associated with it. Of course, we have technologies around to take up phosphorus from, let's say, wastewaters, but still the sludge, which contains all the phosphorus which is taken from the wastewaters, can be posted or buried in landfills, and still there are irreversible loss of phosphorus. For example, I learned that here in Israel, sludge from wastewater treated plants is toasted with wood chips, but still sludge cannot be applied directly to the soil. And I'm a little bit ahead of myself, I would say that there are many pathways of phosphorus loss which can be intercepted with algal cultures. So this is emerging vehicle of phosphorus biocapture, which can possibly solve the problem in the future. Just I would like to show you a few photographs of phosphorus polluted sites. One of them north of Russian Federation, the rock phosphate mine. Another one has been taken not far from here, near the Dead Sea. And there are a lot of biofilms, biocrusts, many structures which contain microalgae. And this is a bright field microscopy showing you the presence of these organisms. When you stay with DAPI, chemical stain, you can see on the fluorescence microalgal cells stuffed with phosphorus result. Complex with DAPI and polyphosphates emits these characteristics green-yellow fluorescence. So, why microalgae? Indeed, microalgae have a formidable potential for phosphorus biosequestration because they're naturally adapted to take up phosphorus whenever they get it. And this phenomenon was called luxury uptake of phosphorus. And algae also generate biomass, which is suitable for diverse applications, which we are already spoken about, from feed, food, till fine chemistry, raw materials. Added benefit is CO2 biocapture and, of course, biofertilizers. But still, we are heavily limited by the lack of basic knowledge about the phosphorus uptake by microalgal cells, especially in the phosphorus eutrophicated. This I will skip to save some time. The main storage pool of the microalgae is polyphosphate. When you starve algae of phosphorus, have normally very low polyphosphate level in the cells, and the total phosphorus content below 1%, whereas in the normal phosphorus sufficient cells is something between 1 and 2% of dry weight. And when you refeed the phosphorus hungry cultures with the total phosphorus in the cell can exceptionally high. It can reach 5, 6, or even 7% of dry weight, but only for the short time. Then the culture resumes division, and the reserves are being mobilized. After the onset of stationary phase, the polyphosphate and other cell reserves reaccumulated. But here comes the first caveat to this, because not all macroalgae are resistant to the sudden increase in phosphorus concentration outside the cells. So there is a phenomenon of sudden macroalgal cell death, which we believe stems from the formation of short chain polyphosphate, when the cell is not able to store it safely in a short time. And this polyphosphate interferes with let's say, protein folding, matrix synthesis process, and many other things. So these are really very important, but you have to be extremely careful with selection of microalgal strains for these processes and for the timing and amount of re-feeding phosphorus to phosphorus-hungry algal cultures. And this transient polyphosphate accumulation was very nicely visualized with Raman microspectroscopy. Scanning mode has been done by Dr. Pedro from Charles University in Prague, hopefully following us by Zoom. And you can see that this pseudo color, blue, these represent polyphosphate, which appear for some time, and it's being mobilized. This, above everything else, is a quantitative approach, which is very nice, maybe not for the field, but definitely for the lab. Okay, and here we keep the size based on our ultrastructural data and some other data was by us and published in the literature, that there is a multi-enzyme, quite complex machine, which is making polyphosphate chains, wraps it in polyhydroxybutyrate sheath. It makes structures which affect very much like a multi-wire electrical cable, OPD wires. Middle isolation, which is polyhydroxybutyrate sheath, something like this. What to do with all this knowledge on kinetics of polyphosphate formation and stabilization? Essentially, it would be used for combining and composing into technologies for efficient biocapture of phosphorus from wastewater. For example, we can use phosphorus-starved cultures for rapid uptake and wastewaters of large amounts of phosphorus. Then they can allow the cells to further divide and grow and to take up more phosphorus, but they obtain higher biomass, but with a lower polyphosphate content. Then they can wait a little bit more and wait when the cells will reaccumulate polyphosphate. Then they obtain essentially what is a good raw material for friendly biofertilizer. We also tested some mobilized algal cells models, which are immobilized chitosan-based cell carriers. This is interesting because mobilization of the cells promotes polyphosphate formation and also solves, to a certain degree, the problems of reducing cell washout from the photobioreactors and biomass harvesting as well. And still, there is another the necessity of adequate supply of inorganic carbon. So to put it another way, you should supply enough CO2 to prevent H from going to alkalinic, because otherwise the phosphorus there becomes non-bioavailable to algal cells. So this is very important to look about the pH and CO2. Otherwise, they are performing very well. And another problem is how to utilize this biomass. I said several times already that it could be biofertilizers. And then we need to answer two major questions. Do the microalgal biofertilizer affect soil health? And are they able to support crop performance? The first question is answered by us and also by the group which works here, led by Livney's participation. As a colleagues from here, the answer is no, it does not disturb soil health. Actually, it's even beneficial for soil health. And there is no deterioration or displacements of soil microbiome, which is a major component of soil health. It's very efficient. So this conclusion is valid for temperate areas and for arid areas as well. Other questions was answered by two large projects, which was done in Germany in collaboration with our university. So in short, it was concluded that algae can indeed carry to the soil and then to the plants sufficient amount of phosphorus, which is comparable to that provided by traditional chemical fertilizers. And this was be done for several crops: wheat, bean, potato. So very important. And what should I say in conclusion? Microalgae are really promising vehicle for wastewater biocapture of phosphorus. And this phosphorus-rich biomass of microalgae, which is produced, can environmentally friendly, slow-release phosphorus biofertilizer, which can be used for different crop cultures. And still, there are a lot of questions to answer. We need really to learn how to control luxury phosphorus uptake by varying the parameters of cultivation. And we need modern economics data to understand how all this works. To be capable of control of this. We need to decide optimal microbial communities, because large biotechnology is story of communities, not of clean cultures. Okay? And indeed, we know how to integrate these microalgal technologies into technologies of wastewater treatment. Just to finish, here's my PhD student working on wastewater treatment plants to learn what should be the best consortium for phosphorus uptake. So that's a real life. And thank you very much. Very successful for us.
Thank you very much. Just the perfect timing. And so we have time for questions, possibly from students, right? Yes, Victor, yes. Yeah, please ask. Yeah.
Okay.
Loudly.
This is a very interesting topic, because I'm also being involved in the same. I just have maybe 3 questions. I try to summarize them about how to starve these cells. Like, how do we starve these cells before you produce them to be able to capture the phosphorus within the hours that you have displayed?
Let me answer one by one.
Okay.
So the answer is you should limit your culture only with the lack of phosphorus. And otherwise, the growth should be without limitation. The only limiting factor is phosphorus. There should be plenty of nitrogen, plenty of light, plenty of everything except phosphorus. Then the cells should divide until they exhaust all their reserves of phosphorus. It takes time because they have polyphosphate inside, in RNA and DNA and many things which can salvage for phosphorus. So this is the approach.
The second question was on how to prepare this biomass fertilizer before you apply it, because there is maybe...
That's excellent questions. I anticipated it. The advantage of biofertilizer from microalgal biomass is that biomass itself is ready-made biofertilizer. You don't even have to till it. You can just throw it on the soil. That's it. The velocity of the release of phosphorus from that biomass is roughly the same the velocity or rate of phosphorus uptake by plant roots. Here you are overcoming the shortcoming of chemical fertilizers, which are rapidly dissolved and fall down into the groundwater. And this is not the case for algal biofertilizer. Experiments have been conducted in Ocean Centrum, so the rate of transfer of radio-labeled phosphorus from algal biomass wheat roots, roughly the same rate of. This is a good thing for us.
Then the last question is, okay, nice.
Okay. We had another question. Wanted to ask a question? Right?
I wanted, yes. What concentrations of phosphorus in the water is good for? Is it high concentrations, or you can?
There are 2 parts in this story. The first part is a real life wastewater. There, in treated wastewater, concentrations of phosphorus are low, below 5 milligrams in liter. And still, this is too high to just discharge wastewater into the environment. And microalgae does a good job of taking it down to the level which is allowed for discharge. Another part is high concentrations, which are not realistic, but can exist in wastewater from rock phosphate mining sites. Still, microalgae can really take down levels to, of course, not to zero, but with time. And their concentration, say 100 milligram per liter.
Want to incorporate the algae in the activated sludge process, like in the same bioreactor of the bacteria?
Again, two ways possible. The first way is to use microalgae to polish the tertiary effluent. Another way is to use microalgae in the secondary stage, in the air tank, but the limiting thing is light, they need light. But still, they also emit oxygen, which can, it's called photosynthetic oxygenation or photosynthetic aeration, which can really increase the efficiency of organics destruction. So it can be this way or other way, but possible either way you want.
What about the contaminants that are present in water?
Well, of course, you clean wastewater from, let's say, some kind of industrial plant which contain a lot of heavy metals. You do not want to put it into the soil on the field. This is a story about bioconcentration and bioreactor. So you can collect biomass, burn it, or store safely for a time. So of sure, there should be wastewater which are more or less safe. For example, wastewater from food processing, from agriculture. It's a common sense to avoid emerging contaminants or hazardous micropollutants.
And if we consider municipal waters,
it's a difficult question on which I'm working right now. So microalgae can very efficiently destroy stuff like antibiotics, medicines which remain in municipal wastewater. They can only, after they can store heavy metals, they cannot destroy it. So if the municipal wastewater is loaded with medicine, antibiotics, or the kind, it's okay. Can be done. It's industry.
The metals are usually gone into sludge.
If you consider tertiary effluent, it's safe. What do you say?
Thank you. Great timing, and I thank all speakers and all audience who came here to listen. We had great talks here. And thank you also for younger scientists presenting really fast and pushing. Thank you, Jose. Thank you, Yoav. Thank you, Alexei, our invited speakers, and Orr, who contributed very interesting study and showed us another perspective of how we can combine different techniques, important issues.